In this video, we will discuss the in vivo delivery of mRNA polymer lipid nanoparticle based complex. As we have discussed in the previous video, that they have developed enhanced lipid nanoparticle delivery system that can transfect the mRNA into the cytoplasm of the cells efficiently, and they have checked the results in the in vitro system. Now they wanted to check whether this lipid nanoparticle polymer lipid nanoparticle based system can transfect the mRNA in the in vivo system also or not, and with what efficiency. So they did their studies at the transfection at the buccal and rectal using two different polymers that they have optimized in the previous results that we have discussed. So they inject the mRNA using mRNA polymer lipid nanoparticle based uh, at the buccal and at the rectal, and for the control they inject direct mRNA alone, and then they measure the signals at 3 hours, 6 hours, 24 hours and 96 hours. And what they are able to find out is that the signals is significantly higher for this 3 hour, 6 hours and 24 hours compared to its respective mRNA alone at 3 hours, 6 hours, 24 hours. And this conditions and this uh, difference is also true when the mRNA is injected at the rectal site using different polymers. So, but at 96 hours they can see the mRNA is again goes down. So this indicates that this mRNA delivery system does not disturb their genomic DNA. At the same time, they enhance the transfections, and in this case they use luciferase firefly mRNA for their studies. So this in this case they you can see that the mRNAs are transfected possibly into the cytoplasm that will cause their translations that leads to the enhanced signals. The signals are highest at the 6 hour, that we can see in the panel B, that at the 6 hour the total emissions from the luciferase firefly is highest. After that it goes down, and at 96 hour it's almost non-significant, the difference between mRNA mRNA polymers lipid nanoparticles based transfection and the mRNA transfections. The similar results they have seen when they transfect the cells at the rectal, that is the signals are highest at 24 hours, but then it goes down, and after at 96 hours the difference is almost non-significant. Even at 24 hours, in this case the difference is non-significant. And they did their similar studies also. They did their similar studies into the using the bioluminescence based mRNA, and in that case they sacrifices the mite at 6 hours, 3 hours, 6 hours, 24 hours, 96 hours, and check the expression of proteins whether it migrates to the different sites or not, as this but and what they are able to find out when they use this lipid polymer lipid nanoparticle based complex, they can see that the transfections of this transfections of their luciferase mRNA is more, and it's reaches to its natural sites, whereas when they use only their luciferase bioluminescence luciferase mRNA, they can see the transfections only at the site of injection. So this further substantiate their findings that substantiate their finding, or this results further made a point that their enhanced delivery of the mRNA using polymer lipid nanoparticle based complex provides a higher transfections efficiency compared to the case where only mRNA is used. And those results are shown in the supplemental figures. You can refer to that paper in order to access that supplemental figures. And so these two experiments further validates that the in vivo deliveries of the mRNA through these polymer lipid nanoparticles based complex has higher transfections compared to the mRNA alone is used for transfections. So I hope you understood how they validate their uh, improved polymer lipid nanoparticle based transfection system. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.